Ayan, full screen na.
Yes, ma'am. Full screen na.
Okay, ayan, sige. Sorry for the technical. Okay. So, naiintindihan naman yung diniscuss ko about compartmentalization.
Opo.
So, with compartmentalization again, it specifically functions for enclosing of the contents of the cell. And then again, you have different membranes like your nuclear and cytoplasmic membranes that do have the specific role. For example, nuclear, more likely for the nucleus, and then for the cytoplasmic membranes, it encloses the other organs or organelles that you see within the cell. And then again, because of this compartmentalization process that's happening within the membrane or the cell membrane or the cell, it's not or the activities within the cell are happening in one place, and it is not being interfered with another activity from other places of the cell. But however, again, these activities of the cell are linked with each other. So for example, if an activity of the cell has occurred in the mitochondria, maybe the next processes like what we have discussed with metabolism, other activities happened also outside of the mitochondria, for example, the cytosol. Okay? And then again, for the other functions, we also have here the scaffold for a scaffold for biochemical activity. So when you say scaffold, it's like a support, okay, for the other biochemical activities happening in the cell. So for example, if you're building a house, of course, for you to be able to build the house, there should be scaffoldings. So these scaffoldings are temporarily or somewhat temporary structures that would actually help you build something. So for the scaffold, it is known that the biological membrane that separates the interior of all cells from the outside environment, which protects the cell from its environment, and it is about 5 to 10 nanometers wide. So that's the size or the thickness of the cell membrane. Okay. So again, even if the plasma membrane is considered a structure that is included in a cell, still it's not just a structure. It plays a role. Okay, so it is included in a biochemical process, and it actually possess proteins and have the capability of doing specific role in the regulation of the cell activities. Okay, so next we also have the function of providing a selective permeable membrane. So membranes prevent unrestricted exchange of molecules from one side to the other. So with this selective permeable membrane, we should always remember that we have what you call the extracellular membrane and the intracellular membrane. So once you say extracellular membrane or the extracellular environment, we are talking about the external side of the cell. So yung mga hindi nakikita sa loob ng cell, that's what you call extracellular. So for the components that you see inside the cell, you call it the intracellular. Okay? So in addition, membranes also provide the means of communication between compartments they separate. So with the membranes, of course, you have the nuclear and the cytoplasmic membranes. These membranes do also have the signaling factors for them to be able to communicate with other compartments of the cell. Okay? So when you also say selective permeable barrier, it means that not all um, maybe not all ions or maybe solutes or liquids can just go in and out of the cell. So there's always a process wherein they follow what type of movement that can be done in your cellular membrane. Okay? And then it is also for transporting solutes. So the plasma membrane contains a machinery for physically transporting substances from one side of the membrane to another, and often from a region of low solute concentration into a region that is at that is much higher in concentration. So you know that concentration gradients are very important when it comes to transporting solutes. Okay, so for example, the presence of salt. We know that the salt is positively charged, and salt is basically a solute. So there's always a gradient that's always being followed. Okay, and then it allows a cell to accumulate substances such as sugars and amino acids that are necessary to fuel its metabolism and build its macromolecules. Okay? So you know that sugars and amino acids are they have a definite size. Okay, so of course there would always be a transporting mechanism that would support this sugar and amino acids going in and out of the cell. And then it is also able to transport specific ions, thereby establishing ionic gradients across itself. So it's very important also that the cell would have a well-balanced intracellular and extracellular environment for it to be able to push through with its activities. So, kahit naman tayo 'di ba? Kapag nawawalan tayo ng energy, we don't really tend to do activities. But if we're full of energy, we tend to do much more things. So, it's just like the cell. Okay? So next, we have responding to external stimuli. So when you say responding to external stimuli, we call it the signal transduction. Okay? So for the signal transduction, membranes possess receptors that combine with specific molecules we call the ligands or respond to other types of stimuli. It can be, for example, light or mechanical tension. So depending on the situation, there are several factors that would affect signal transduction. So light and mechanical tension are just some of the examples that we have for it. Okay? So different types of cell have membranes with different receptors and they are therefore capable of recognizing and responding to different environmental stimuli. So the interaction of a plasma membrane receptor with an external stimulus may cause the membrane to generate a signal that can be stimulatory or inhibitory for the internal activities. Okay? So depending on which or on what type of stimulus that is being received by the cell, of course, it can be a stimulatory process or activity. So it means na pinapabilis niya or nagdadagdag siya ng mga activities na pwedeng gawin ni cell, or it can be inhibitory. Pag sinabing inhibitory, i-stop niya ang isang process ni cell. That's to be able for the cell to have a balanced environment. Okay? So next, we have intracellular interaction. So the plasma membrane allows cells to recognize and signal one another and to adhere when appropriate and to exchange materials and information. So that's intracellular interaction. So this interaction is more likely on the intracellular matrix of the cell. Okay? So walang kinalaman yung outside ng cell. So proteins within the plasma membrane may also facilitate the interaction between extracellular materials and the intracellular cytoskeleton itself. Okay? So lastly, we have energy transduction. So if kanina we have what you call the signal transduction, in here we have energy transduction. So membranes are intimately involved in the process by which one type of energy is converted to to another type. So actually, the most fundamental energy transduction occurs during photosynthesis. So if you recall what happens in photosynthesis during this process, the energy from the sunlight is absorbed by the membrane-bound pigments, and then they are converted into chemical energy and then is stored in the form of carbohydrates. Okay, so that's how important energy transduction is. So if you also go back to the previous lectures that we have learned already how energy transduction happens. So it's the change of one form of energy to another. Okay, and then membranes are also involved in transfer of chemical energy from carbohydrates and fats to the ATP. Okay, so we all know that carbohydrates and fats are able to produce ATP for us to use. Okay, so basically, ang pinakaunang nauubos talaga, for example, in a human body is the carbohydrates. And then eventually, if carbohydrates are no longer available, we have your fat lipids. So they, in exchange of the carbohydrates, the fats are being burned to for us to be able to have energy. And then eventually, kapag wala na, of course, ang huling-huli na lang na maaapektuhan dyan ay yung mga proteins. Okay? So for the chemical compositions of the membranes, so the first in here is that you should know that membranes are lipid protein assemblies. So there's always a combination of fats and proteins. So in which the components are held together in a thin sheet of non-covalent bonds. So for the lipid layer of your cell membrane, it's actually characterized as a bilayer. Okay, so sabi, dalawang layers daw ang lipid na makikita natin sa cell membrane. So this serves primarily as structural backbone of the membrane and provides the barrier that prevents random movements of water-soluble materials in and out of the cell. As we all know, pag sinabi nating lipids or fats, they are usually hydrophobic to water. So they don't usually mix with water. Okay, so that's basically the essence of having a lipid bilayer. It's for them to be able um, to prevent random movements of water molecules or water soluble minerals as well, leakage of water soluble minerals. Okay. And then for the protein part, each type of differentiated cell contains a unique complement of the membrane proteins that contributes to the specialized activities of that type of cell. So those proteins are actually embedded on the cell membrane. So the cell membrane is not 100% made up of lipid. There are embedded proteins in it. So these proteins are very important when it comes to the biological processes that's happening in the cell. So later on, we will be discussing more about these proteins that you see on your cell membrane. Okay. So for the ratio of lipid to protein in a membrane, it actually varies depending on the following instances. Okay, so first you have the type of cellular membrane. So for example, the cell membrane versus the um, membrane of the endoplasmic reticulum, and then the membrane of the Golgi apparatus. So it is also dependent on the type of organism, may it be prokaryote, may it be plant or an animal. And then, of course, the type of cell. So depending on what type of cell, you have cartilaginous, you have muscle, you have liver, you have blood cells. So depending on what type. Okay. So this image that you see here on your screen right now is a figure representation of the plasma membranes as lipids plus the proteins. So as you see here, this is the bilipid layer. Okay? So all of the hydrophilic heads are outside and the hydrophobic tails are within or embedded within the bilipid layer. So apart from the bilipid layer, you will also see their cholesterols, phospholipids, integral proteins. So these types of proteins are the ones helping when it comes to the moderation and the movement of particles, ions, and other materials that's going in and out of the cell. You also have peripheral proteins. You also have your glycolipids or oligosaccharides. Okay, that's embedded within your cell membrane. Okay, so for um, the membrane lipids. So membranes contain a wide diversity of lipids, all of which are amphipathic. When you say amphipathic, so they contain both hydrophilic and hydrophobic regions. Okay, so there are three types of membrane lipids. The first one is what we call the phosphoglycerides. So the phosphoglycerides are glycerol-based phospholipids, meaning their backbone or their main strand is made up of glycerol, and they often contain one unsaturated and one saturated fatty acid chain. Okay. And then the next one, we have your sphingolipids. These types are less abundant class of membrane lipids, and they are derivatives of your sphingosine, which is an amino alcohol that contains a long hydrocarbon chain. So sphingolipids are actually considered as ceramides. So when you say ceramides, meaning it's a waxy type of lipid. Okay? So hindi siya yung katulad lang ng, for example, fat na madulas. This type is waxy. So imagine, for example, you have a candle, di ba? The wax there is considered to be it's actually a type of lipid na waxy. Okay, hindi siya basta-basta humahalo sa tubig. Okay? And then next, you have cholesterol. It's a sterol that can be up to 50% of animal membrane lipids. And however, it is missing from most plants and all bacterial cell membranes. So cholesterol is more likely to be found on animals. Okay? So each type of cellular membrane has its own characteristic lipid composition. So the nature of the head groups are actually very important when it comes to um, you know knowing this lipid composition. So they differ from each other in types of lipids, nature of the head groups, and particular species of fatty acid chains. And because of the structural variability of these lipids, it is estimated that some biological membranes contain hundreds of chemically distinct species of phospholipids. So some biological membranes contain more than 100. Okay, so that makes them very distinct. So whenever that the head or the nature of the head group of a certain lipid changes, of course, the conformation or the result of that um, changes would definitely become another type of lipid. So the percentage of some of the major types of lipids of a variety of membranes are given in table 4.1. So as you see here, we have phosphatidic acid, like in human embryos, it's around 1.5 in the given total weight percent of a total lipid. So they also have phosphatidylcholine, etc. Okay? So those are some of just the lipid compositions of some biological membranes. Okay? So next, so the lipids of a membrane are more than simple structure elements, but they can have important effects on the biological properties of a membrane. So in here, one, you should determine the physical state of the membrane. Two, it can influence the activity of a particular membrane protein. And third, it can provide precursors for highly active chemical messengers that can regulate cellular function. Okay, so those are this even if it's a simple structure, but then again it has numerous roles that needs to be um, put into consideration when it comes to the effects on biological properties. Okay, so next, combined fatty acid chains of lipids span width of are approximately around 30 angstrom, and each row of head groups adds another 15 angstroms. So thus entire bilayer is only around 60 angstroms or 6 nanometers in thickness. Okay? And then you also have membranes do not have free edges or ends and are always continuous and unbroken. So membranes are never seen to have free edges, and it is because membranes form extensive interconnected networks within the cell. So there's a flexibility of the lipid bilayer, and membranes are deformable, and their overall shape can change as occurs during locomotion or cell division. So wala pa naman kayong nakitang ano, 'di ba? Examples ng cell na broken sila. Usually they are continuous, they are bounded. Um, the effects of the of ions or some molecules to go in and out of the cell doesn't really affect the integrity of the cell membrane. Okay, so it's always continuous, and it has actually the capability of self-assembly. So when you say self-assembly, it can put back itself together. Okay, so that's one of the important functions, or one of the important features of your cell membrane. Okay, so the lipid bilayer is also thought to facilitate regulated fusion or budding of membranes and self-assembly. Okay, so it has its own ways of being able to put back itself together. So next, let's talk about the membrane carbohydrates. So after the lipids, let's go to the carbohydrates. So in here, eukaryotic cell plasma membranes also contain, again, your carbohydrates. So all carbohydrates of the plasma membrane faces outward into the extracellular space. So if this is the cell membrane or the bilipid layer, more of the carbohydrates are facing outwards, okay, into the extracellular space. So the carbohydrate of the intracellular membranes also faces away from the cytosol. So if this is the cytosol's side, all of the carbohydrates are on the other side of it. Okay. So for the modifications of your carbohydrates, so the carbohydrates of your glycoproteins is present as short, branched hydrophilic oligosaccharides, and typically having only less than 15 sugars per chain. Okay. And then for glycolipids, or the carbohydrates of your glycolipids like your blood, they are most exposed structures on the extracellular surface of the cells and are flexible with numerous binding sites, which make the optimal for cell signaling. So for the addition of carbohydrates in your may it be for proteins or lipids, you call it through the process of glycosylation. Okay, so when you add carbohydrates, you you undergo the process of glycosylation, and it's actually one of the most complex processes happening within the biomolecules. Okay, so in here you will see just a representation of how your carbohydrates do actually look like when they are, for example, linked to glycosphingolipids. So they are short as well. For example, the other example are for glycoproteins. So that's how their carbohydrates look like. Okay. Next, let's go to membrane proteins. So depending on the cell type and the organelle within that cell, a membrane may contain hundreds of different proteins that has a defined orientation relative to the cytoplasm, making the properties of one surface of a membrane very different from those of the other surface. Hence, this is a asymmetry, which is referred to as membrane sidedness. Okay, so for the proteins, like also the carbohydrates, they tend to have a side wherein dun sila present, where they are present. Okay, so for proteins, the orientation is relative to the cytoplasm. So the parts of the proteins interacting with extracellular substances other than cells and extracellular matrix elements actually phase out, and then while those interacting with cytoplasmic molecules are actually facing inward. Okay? So there are actually different classes of membrane proteins. So these classes are distinguished by the intimacy of the relationship to the bilipid layer. Okay, so the first one, we have the integral proteins, which are transmembrane proteins. So when you say transmembrane proteins, they are the ones embedded cell membrane. Okay, so they pass entirely through the bilayer and have domains that protrude from both the extracellular and cytoplasmic sides of the membrane. So some research says that they constitute 25 to 30% of all encoded proteins and roughly 60% of all our current drug targets. Okay? And then the next type or the next class would be the peripheral proteins. So they are located entirely outside of the lipid bilayer on either the cytoplasmic or extracellular side, and then yet are associated with the surface of the membrane by non-covalent bonds. Okay? So hence the name peripheral. When you say peripheral, anything that's away from the midline. So nasa tagiliran sila or nasa gilid sila. Okay? And then the last class would be a lipid-anchored protein. So it is located outside the lipid layer on either the extracellular or cytoplasmic surface and are covalently linked to a lipid molecule that is situated within the bilayer. So hence lipid anchored, ibig sabihin it is a protein that has a linkage with a lipid. Okay? So these are the three classes of membrane proteins. So the first one, again, we have the integral proteins, which are typically or do typically contain one or more transmembrane helices. So these are the helices that you see. And then the next one, um, we have your peripheral membrane proteins. So if these are the helices, so the peripheral proteins are not embedded. So they are just on the sides of the bilipid layer. And then you have now the lipid anchored protein. So this one is a protein that is associated or linked to a lipid. As we all know, the bilipid layer, it has a hydrophobic tail. It's a type of lipid. Okay, so that's how it is being linked to it. Okay, so for the integral membrane proteins, we have different functions. So for it actually acts as receptors that bind specific substances at the membrane surface. So it also has channels or transporters involved in the movement of ions and solutes across the membrane. Or it can also be an agent that transfer electrons during the process of photosynthesis and respiration. So for this integral membrane proteins, it's not only atoms or particles, charged particles that can be transported in and out of the cell, but these integral proteins can also help when it comes to the passage of solutes, so bigger types of molecules. Okay? And then its characteristics, again, these integral proteins are amphipathic. So again, they do have the uh, hydrophobic and hydrophilic ends. And then portions of an integral membrane protein that reside within the lipid layer or the transmembrane domain tend to have a hydrophobic characteristic. Okay? And then portions of an integral membrane protein that project into either the cytoplasm or extracellular space tend to be more like the globular proteins to have hydrophilic surfaces. And hence with this one, they interact with water-soluble substances like low molecular weight substrates, hormones, and other proteins at the edge of the membrane. So anything that's, in the middle of the bilipid layer, and of course the portion also of the integral membrane, more likely they are hydrophobic in nature. But then the free edges or the edges that you see outside of the cell and then through the cytoplasm, more likely they are hydrophilic surfaces. Okay? So in this one, the historical background of the discovery of the integral membrane proteins were actually through the freeze fracture replication analysis. Okay, so this technique was used to prove the concept that proteins penetrate through membranes. Okay, so again, that's what you call the freeze fracture replication analysis. Okay. So if you read your book, it's actually there. The history and the process of the freeze fracture replication is indicated in your book. Okay. So next, let's go to the peripheral membrane proteins. So it is associated with the membrane by weak electrostatic bonds, and then can usually be solubilized by extraction with high concentration salt solutions that weaken the electrostatic bonds holding peripheral proteins to a membrane. So for it to be able to be um, solubilized or yung matunaw, kailangan niya ng mga high concentration of solutions. Okay? So the best studied peripheral proteins are located in the internal or cytosolic surface of the plasma membrane, where they form a fibular network that acts as a membrane or skeleton. And these proteins provide mechanical support for the membrane and function as an anchor for integral membrane proteins. And then other peripheral proteins on the internal plasma membrane surface function as enzymes, specialized coats or factors that transmit transmembrane signals. Okay? So for the lipid-anchored membrane proteins, there are actually several types of lipid-anchored membranes that can be distinguished, and numerous proteins present on the external face of the plasma membrane are bound to the membrane by a small complex oligosaccharide linked to a molecule of phosphatidyl linositol that is embedded in the outer leaflet of the lipid bilayer. And then peripheral membrane proteins containing this type of glycophosphatidyl linositol linkage are called your GPI anchored proteins. So once they do have this phosphatidyl linositol, it is already called a GPI anchored protein. So for us to be able to identify transmembrane domains, the question that we can have in mind is which segments of the polypeptide chain are embedded in the lipid bilayer. So when you say transmembrane domains, these are segments of a protein embedded within the membrane. So they have a simple structure consisting of a string of about 20 predominantly nonpolar amino acids that span the core of the lipid bilayer as an alpha helix. Okay? So they tend to form alpha helix here. So an example that we have here is glycophorin A, which is an integral protein with a single transmembrane domain. So, so this is actually, or this glycophorin A is actually a major erythrocyte cell membrane integral protein. So the chemical structure of a single transmembrane helix is shown here on this image right here, which depicts the two-dimensional structure of glycophorin A, the major integral protein of the erythrocyte plasma membrane. Of the 20 amino acids that make up the lone alpha helix glycophorin monomer, amino acid 73 to 92 of this figure, as you see my figures dyan, 90 and then 80. So, yung nasa gitna nun, mga amino acid 73 to 92, uh, all have but three hydrophobic chains. So all of them, um, among those, three only have hydrophobic side chains. Okay, so the exceptions are serine and threonine, which are non-charged and polar residues. So it is noted that alpha helix is favored conformation because it allows a maximum number of hydrogen bonds to be formed between atoms of polypeptide backbones, thereby creating a highly stable low energy configuration. Okay, so this is or having an alpha helix conformation is easier than, of course, the proteins which are tertiary or quaternary in structure. Okay, so knowing the amino acid sequence of an integral membrane protein, we can usually identify the transmembrane segments using a hydropathy plot. So this is where each site along a polypeptide is assigned a value that provides a measure of the hydrophobicity of an amino acid at that site as well as that of its neighbors. And then this approach provides a running average of the hydrophobicity of short sections of the polypeptide and guarantees that one or few polar amino acids in a sequence do not alter the profile of the entire stretch. And then hydrophobicity of amino acids can be determined using various criteria such as their lipid solubility or the energy that would be required to transfer them from a nonpolar medium into an aqueous medium. Okay? So, determining spatial relationships between amino acids with integral membrane proteins. So, you will be able to know this interrelationship between these two by first the use of site-directed mutagenesis. So, this is by the process of introducing specific changes into the gene that encodes the protein. So, as we all know, proteins do have their own um, gene encode, gene profile. Okay, but then again, if you tend to or if you introduce a different change or a different gene on a specific site of that protein, of course, it's what you already know as the mutation process. So by introducing a different type of gene, of course, there would be a different result also of what type of protein will be produced. Okay? And then the next one, we can also clarify dynamic events occurring as protein functions. So this is by introducing chemical groups whose properties are sensitive to distance between them, which results to the activation of the channel that is accompanied by an increased separation between the labeled residues of the subunits. So, ganun din pala, no, kapag dating sa mga amino acids and integral membrane proteins, meron ding kinalaman ang distance. Para siyang relationship, no? Parang hindi siya gumagana pag LDR kayo, pero kapag ka magkalapit kayo, more likely it's the partnership is activated. So that's the concept for you to be able to easily understand how these amino acids and integral membrane proteins are interrelated or how can they work together. Okay, so distance is a factor for them. Okay. So next, for the membrane lipids and fluidity. So the physical state of the lipid of a membrane is described by its fluidity or viscosity. So fluidity is actually the measure of ease flow, and viscosity is the measure of resistance to flow, which are actually inversely related. So you know that plasma membranes, they are very soft, they are very fluid. Kaya nga, 'di ba, even if there's too much locomotion or there's too much tension or movement, they don't really break easily. It's because of this characteristics of having fluid. It's a fluid filled mosaic structure, and of course, there's always viscosity. Okay, so lipids exist in two states. So it can be solid and in the liquid phase of varying viscosity depending on the temperature. So for example, we all know that, for example, if a cooking oil is placed in the fridge, there are types of lipids or cooking oil that when you put them in the fridge, they tend to become solid. But if you put them in a regular room temperature, it becomes liquid again. So again, temperature is another factor for you to be able to consider the membrane fluidity of lipids. Okay, and then transition temperature is actually the temperature at which membrane goes from fluid state to crystalline gel. So there's always a transition phase for lipids to, to go back to which state they would like to be in. So depending again on the temperature. So what are the importance of membrane fluidity? So first, membrane fluidity provides a perfect compromise between rigid ordered structure and a totally fluid, non-viscous liquid. And then moderate fluidity also allows interaction to take place within the membrane. Clusters of membrane proteins can assemble at particular sites within the membrane and form specialized structures. And then membranes arise only from pre-existing membranes. So their growth is accomplished by the insertion of lipid and protein components into the fluid matrix of the membranous sheet. And many of the most basic cell processes like cell movement, growth and division, intracellular junction formation, secretion, endocytosis, and exocytosis depend on membrane component movement. So imagine if the cell is very rigid, no? Paano kaya gagalaw yung mga organelles sa loob? Paano kaya how will it be able to be how will it be possible for water to move in and out of the cell just easily if the cell is very rigid? So we all know that this fluidity and flexibility of the cell membrane really helps when it comes to you know uh, having a balanced activity within the cell. Okay, it's not only for the biochemical processes, but of course for the movement of the different organelles of the cell as well. Okay. So next, maintaining membrane fluidity. Cells respond to environmental changes in temperature by regulating membrane fluidity, except for birds and mammals, which are warm-blooded. So usually homeostasis at cellular level is being used. Okay? And then next, asymmetry of the membrane lipids. So lipids are distributed in highly asymmetric pattern, some more prevalent in the outer leaflet and others on the inner leaflet. So when you say leaflets, so these are two halves of the bilipid layer. You have the cytoplasmic leaflet, and then you also have the exoplasmic leaflet, which actually faces outside the cell or into an organelle. So yun lang yun, the intra and the extracellular matrices. So those are just the leaflets. Okay. So next, we have here the dynamic nature of the plasma membrane. So we have the lipid and protein mobility process. So lipid bilayer is relatively fluid, as we have discussed. So the mobility of individual lipid molecules within the bilayer of the plasma membrane can be directly observed under the microscope by linking the polar heads of the lipids to gold particles of fluorescent compounds. So in this one, with the experimental demonstration that diffusion of phospholipids within the plasma membrane is confined. So from here, no, once the mobility already happens or the movement already happens, you will see that there can be differences in the conformation or with the direction of where this lipid and proteins of the plasma membrane goes to. Okay, so in here for the figure 4.25, you will see that the possible movements of phospholipids in a membrane. So pwede siyang lateral shift, pwede siyang paikot, pwedeng papaloob. So depende. Okay, so some of them also flexes. Okay, there are different ways or modes of mobility. So next, the diffusion of membrane proteins after cell fusion. So when you say cell fusion, it is a technique whereby two different types of cell or cells from two different species can be fused to produce one cell with a common cytoplasm and a single continuous plasma membrane. Okay? So cells are induced to fuse with one another by making the outer surface of the cells sticky so that their plasma membranes adhere to one another. So cells can be induced to fuse by addition of certain inactivated viruses that attach to the surface membrane by adding the compound polyethylene glycol or by a mild electric shock. So cell fusion has played an important role in cell biology and is currently used in an invaluable technique to prepare specific antibodies. Okay, so usually these cell fusions um, marami nang pinagamitan tong cell fusion na to. Like for example, GMOs, 'di ba? For example, if you would like um cross-link different types of species, if you would like to produce crops na gamit na tong cell fusion. So pwede din siya sa IVF, 'di ba? Sa IVF or kapag when a couple can't have babies, they use the concept of cell fusion for IVF. So, ayan. So, it is now also being used for the development of certain drugs or antibodies. Okay? So, for cell fusion, the first experiments to demonstrate that membrane proteins could move within the plane of the membrane utilized cell fusion were actually reported in 1970 by Larry Fry and Michael Edidin of Johns Hopkins University. So this image that you see here on your screen right now is the use of cell fusion to reveal the mobility of the membrane proteins. So for the outline of the experiment in which human and mouse cells were fused, so steps 1 and 2, ito yung ginawa nilang fusing. You have the human cell and then the mouse cell, and then the addition of Sendai virus or the fusing virus. Ganito yung nangyari. So, nag-fuse sila. And then next, the distribution of the proteins on the surface of each cell were followed in the hybrids over time, which are already, which is shown here on image 3 and 4. Okay? So, mouse membrane proteins are indicated by solid circles, human embryo proteins by the open circles. So locations of the human and mouse proteins in the plasma membranes of the hybrid cells were monitored by interaction with fluorescent red and fluorescent green antibodies, respectively. So in here, with the colors that you see, you will now um, definitely can trace where the different proteins of the different cells actually went to or directed. Okay? So, nagkalat na sila sa loob after. Kaya kapag tinignan natin yung, or if we check on the fluorescent image, ganyan siya. You see the green parts. So, sabi sa green fluorescent, that's the antibodies for um, the mouse, and then the red ones are for the antibodies or fluorescent antibodies of the human cell. Okay, so those are the proteins. Okay. So protein mobility patterns in living cell membranes are shown by two light microscopy methods which are excellent for measuring protein movement, extent, and rate. So apart from the experiment of Fry and his colleague, we can also use different techniques for us to be able to check naman on other types of cell fusions or the movement and the rate. Okay? So it's not only the direction that we are looking at, but of course we are also looking at the rate or the time wherein the fusion of these cells are effective or gaano ba katagal yung nangyayari dun sa gaano ba katagal yung pag-move ng mga proteins na yon. Okay? So the first one, we have the FRAP or the fluorescence recovery after photobleaching. So this technique is a technique wherein you use fluorescent dyes and bleaching. Okay? So first, a particular component of the membrane is labeled with a fluorescent dye. And then after that process, a small region of the surface is then irradiated to bleach the dye molecules. And then the third is the recovery of fluorescence in the bleach region is followed over time. So it's very important that um, when tracing the mobility of lipid and proteins, time is really a factor. Okay? And then the second one is the single particles tracking or SPT. So individual membrane protein molecules are labeled and usually with fluorescent molecular tags that emit light under a microscope. So those are the two other techniques being used. And then restraints on protein mobility or factors affecting membrane protein fusion. In summary, includes lipid matrix viscosity and protein mass are partially responsible, but other forces also restrain protein mobility. So there's always, factors that could restrain or could stop this mobility of the proteins. Okay? So for the control of the membrane motility, so there are interactions occurring within the membrane itself and materials on outer surface control membrane motility. So membrane lipid mobility, phospholipids are small molecules that make up the very fabric of the lipid bilayer, and one would expect their movement to be unfettered, but they also seem to be restricted. So when you say unfettered, guys, it is not really controlled most of the time. It cannot be restricted. So, um, ganon ang lipid mobility. Okay? For the membrane domains and cell polarity, so most studies of the membrane dynamics were carried out on the relatively homogeneous plasma membrane situated at the upper or lower surface of a cell residing on a culture dish. However, results exhibit distinct variations in protein composition and mobility, especially in cells whose various surfaces display distinct functions. Okay? So for an example for the plasma membrane structure, we had your red blood cells. So why is RBC membrane so well studied and best understood membrane? So the reasons are the following. So the cells are inexpensive to obtain and readily available in huge numbers from whole blood. And then the cells are already present as single cells and need not to be dissociated from a complex tissue. And then next, RBCs are simple in comparison to other cells. They have no contaminating internal membranes. And lastly, one can obtain pure intact RBC membranes by hypotonic lysis of cell hemolysis. Okay? So basically, an RBC doesn't really have a nucleus. If you check on its characteristics under a microscope, it's just a circular, hollow, biconcave. It has a biconcave center, but doesn't really have any organelles in it. So it's really plain and simple. So for the membrane itself, so for you to be able to extract it from or to separate it from the liquids or the um, mixture within the RBC, of course, all you have to do is to have it exploded, okay, for you to be able to check on the membrane of the RBC. So when you say hypotonic lysis, usually um, there should be a solution gradient for it to become a hypotonic, for you to be able to have a um, hypotonic solution. So when you say solution, of course, it requires you to have salt. Okay. So next, SDS, polyacrylamide gel electrophoresis, or SDS page, is used to separate or fractionate membrane proteins after they have been solubilized, and it gives one an idea of the membrane protein diversity. And the next, erythrocyte membrane integral proteins. It is most abundant integral proteins are two protein containing membrane spanning proteins, band 3 and glycophorin A. And again, freeze fracture shows them at high density. And then erythrocyte fibrillar membrane skeleton is made up of peripheral membrane proteins on inner surface and maintains a biconcave RBC shape under punishment of circulation and restrains integral protein movement. Okay? So for the movement of substances across cell membranes, so let's go now to the different cell movements, like for example, diffusion and osmosis. So membrane has actually dual function. First, it retains dissolved materials of the cell so they do not leak out of the environment. And second, it must allow the necessary exchange of materials in and out of the cell. Okay. So energetics of the solute movement depends on the magnitude of the concentration gradient. Okay? And when solute is a non-electrolyte, it will move down a concentration gradient or exergonic. Okay, when you say non-electrolyte, these are the atoms that doesn't really have or that are negatively charged. Okay? Kasi kapag ka-electrolyte, those are the ions that have or do have electrical charges. Okay? So if solute is an electrolyte, the overall charge difference is also very important. So you should also take note of the potential differences in voltage and bigger difference in charge. Of course, there will be a bigger difference in free energy. So diffusion of substances through membrane. So there are two qualifications that must be met before non-electrolyte can diffuse passively across a membrane. So first, substances must go down a gradient. So must be present at a higher concentration on one side of the membrane than the other. So, and the next membrane must be permeable to substances. So when you say passive diffusion, guys, when you hear passive, it just means that it doesn't really require ATP or energy for a certain molecule or for a certain um, substance to go in and out of the cell. So it just goes freely. It can pass through the cell membrane without energy being required. Okay? But when you say active diffusion or active transport, it would always require you to have energy. And of course, electrolytes or those ions that do have electrical charges. Okay? So next, membranes are permeable to a given solute in two ways. So solute can pass directly through bilayer or solute can traverse an aqueous channel or pore that spans the membrane and prevents the solute from coming out contact with lipids of the bilayer. Okay? So other factors that determine the ability of molecules to pass directly through the membrane. First is the polarity of the solute. So this is a measure of polarity or non-polarity. It is a partition coefficient. So when you say polarity, diba, this is it refers to the electrical conditions determining the direction of the current flow relative to the electrode. And then the size, of course, if it's a small molecule, it can pass through the membrane faster, and small inorganic substances penetrate rapidly, and bigger polar molecules do not pass easily or not at all. Okay? And then diffusion of water through membranes. Since water moves faster through a membrane than solutes, so membranes are called semi-permeable. Okay? Kaya siya, because of this characteristic, dyan nakuha ni cell membrane yung tinatawag na semi-permeable membrane. Okay? So in here, you will see the four basic mechanisms by which solute molecules move across membrane. So again, you have the passive and then the active. So for passive non-mediated processes, so A is actually a simple diffusion. This happens with the water. For B, simple diffusion for that has a channel. Diba? You have the integral protein. So some integral proteins are just open. Open-ended sila. Open sila sa labas, open sila sa loob. So this channel or this pore can actually let molecules or substances pass through it. Okay? And then the example is actually salt. Okay? And then for transporter mediated or the mediated type of transport in the cell, you also have letter C here, which is the facilitated diffusion, wherein which solute molecules bind specifically to a membrane protein carrier. So in here, if this one is just a a free open-ended type of protein, for transporter or facilitated diffusion, there should be a specific protein that would work for a specific substance. So dito sa part na to, pagka sinabing facilitated diffusion, kailangan complementary yung integral protein and yung substance. Hindi pwedeng basta-basta na lang papasok yung substance kung yung protein naman na yun ay hindi compatible para sa kanya. Okay? So for example, for glucose, there should be a complementary glucose integral protein for it to assist the glucose molecule to go in of the cell. Okay, that's what you call a facilitated diffusion. Okay, and then next, you also have the active transport by the means of protein transporters, which wherein there are specific binding sites that undergoes change in affinity driven with energy released by an exergonic process. So in here, the example would be, of course, if the ATP would actually bind to a position of the integral protein, kumbaga nagbibigay siya ng energy dun sa protein na yun to open and close the channels. Okay? So without the proper energy or maybe electrolyte that would stick to the integral protein, the channels won't open. Okay? May mga tinatawag tayong mga receptor binding sites pa. Okay? When you say there are receptor binding sites, the receptors can only open certain pores wherein kailangan yung marireceive nila ng mga substances or ions are compatible with them. Okay? So, pwede partly sarado, this part will be closed, and then eventually if ATP will be introduced on that particular integral protein, it will open. Okay? Once it's open, the ions can go through, and then once na nakapasok na, eventually the other part will close and then the other end will open. Okay, so that's one example. So the best example that I could provide you for this active transport with the use of a specific protein transporters is the sodium and potassium ion concept. Okay, so usually these are being this type of transport system is used for muscles, for the relaxation and for the constriction of muscles. 'Di ba if you have cramps, ibig sabihin lang nun constricted yung muscles, 'di ba? So all you have to do is to take in potassium, which is more likely being consumed through banana. So si banana, since it has more potassium, when you eat it, so si potassium pumapasok ngayon dun sa cell. Okay? So sa cell na yon, pag pumasok, when potassium ions go in the cell, on the other hand, sodium ions should go out of the cell because sodium ion is a type of an electrolyte. And an electrolyte actually gives you energy. So when there's energy, there's continuous constriction of muscles. So kailangan magpalit sila ng position. There should be an exchange of position. So when three sodium ions go out, there should be two ions of potassium that should go in of the cell para bumalik sa homeostasis yung katawan. So, to prevent further constriction of your muscles. Okay? Nagigets ba? Okay? So, more likely may kinalaman talaga ang mga charges also. And of course, the number of this electrolytes in your cells. Okay? So, ganun ang mga, those are the mechanisms across the, or those are the different transport processes that could happen in your membrane. Okay? So, next, osmosis is an important factor in the multitude of bodily functions. So, digestive tract secretes several liters of fluid daily, and it is reabsorbed osmotically by the cells lining the intestine. If it is not reabsorbed, as in cases of extreme diarrhea, it can lead to rapid dehydration. Okay, so this is also an example that I could give you is that, for example, if you have diarrhea, is it okay to drink Gatorade? Do you think that Gatorade would really suffice your diarrhea? Okay, so when you have diarrhea, your intestinal muscles are not constricted. Okay, more relaxed sila. Kaya nga kapag may diarrhea, tuloy-tuloy ang pagdaloy ng ginhawa. Pero minsan sa sobrang pagdadiarrhea mo na, hindi na siya maganda sa katawan. So what you have to do is to stop this relaxation. No? So basically for diarrhea and the energy drink, you know that energy drink has a different component. Energy drink are actually or are usually used only for athletes. They are designed for athletes but not for diarrhea. So for diarrhea, it's more likely that you should use or you should drink electrolyte-rich drinks. Like, for example, Pocari Sweat. Si Pocari Sweat, it's a little bit salty. The salt would actually help you have constriction of the intestinal muscles, just like what I've explained to you with your basic constriction and relaxation of muscles. Okay, so the more you drink, of course, electrolytes in your body, of course, the cell would eventually adjust to the number of sodium ions that you have in your body. Okay? But then again, Gatorade do have a different or does have a different mechanism. It's not really for diarrhea because 'di ba if you're an athlete, you are always sweating, right? So if you still drink or electrolyte rich drink, so kung mas marami kang salt sa katawan, 'di ba ang ginagawa ng salt sa mga cells ay kapag ka nadadamihan ay nagsishrink. So ang mangyayari lang niyan if you continue to drink sodium rich na mga drink for athletes, syempre yung cells mo mauubusan ng water sa loob yan. Puputok yan. Lalo kang mapapagod. So yung components of Gatorade is actually not the same as the components of your Pocari Sweat. And they don't really have the same mechanism of action in your body. So for Gatorade, of course, yung mas kailangan ng katawan, yun yung kailangang i-provide nung drink. So kung masyado nang pagod ang isang athlete, iinom ka pa ba ng mas marami pang salt? 'Di ba hindi naman? Okay? So that's how actually osmosis happens. Okay? So there's always um, an incorporation of the idea of solutions, of salt. Kailangan may salt. Okay? So usually with osmosis, where sodium ion is, water follows. So kung saan mas mataas or where the sodium gradient is higher, water always follows there. Okay? Mas gusto ni water na dumikit sa mga sodium ions for osmosis, in the idea of osmosis. Okay? So naiintindihan naman? Okay pa naman? So those are just basic explanations or examples for you to be able to easily understand the concepts of osmosis and diffusion. Okay? So next, plant cells are usually hypertonic compared to the fluid environment. So when you say hypertonic, mas mataas yung gradient or the gradient of the salt is higher. Unlike in animal cells which are isotonic. So when you say isotonic, the internal and the external matrix or matrices should be equal. Okay? And then water tends to enter the cell causing internal turgor pressure to push against the cell wall. So for pag hypertonic naman, ay kapag hypotonic naman, yun naman yung masyadong mataas naman yung salt solution sa loob ng cell, wherein yun nga kapag ka ang cell mas maraming salt, pumapasok si water. So pag marami ng water sa hypotonic solution, ang cell nagbo-burst. Okay? So many cells are much more permeable to water than can be explained by simple diffusion through the bilipid layer. So this is the example of hypotonic, hypertonic, and isotonic solution, and the effects of the differences of the concentration of solutes on opposite sides of the plasma membrane. So I hope that you already know this concept. This is very basic. So again, when hypotonic, so basically, mas mataas or the gradient of the salt is larger or higher inside the cell. Of course, the water would all go in. Eventually, for a cell when when it is in a hypotonic environment, it will eventually burst. And then for hypertonic, since the salt is more likely higher outside, of course, all of the components of the cell would go out. Okay, so eventually the cell will shrink. But for an isotonic solution, since the molecules of or the gradients in and outside of the cell are equal, so there is no net loss or gain. So so the integrity of the cell will just be intact. Okay? So diffusion of ions. So ions go through ion channels like the integral proteins surrounding the aqueous pores. Since core of the lipid bilayer is highly impermeable to charged substances like sodium, potassium, calcium, and chloride. So apart from, we have, di ba nga, we have the integral protein. So some of them are also considered ion channels. So those ion channels, sila nga when there is a um, incorporation or pag dumidikit yung um, mga ions na yun, tsaka sila gumagana. Okay? So, next, bewildering variety of ion channels identified, each with integral proteins surrounding aqueous pore, and most are highly selective in allowing one particular type of ion to pass through a pore. So again, there are ion channels that are very, very particular with which type of ion they can assist, okay, with the diffusion of ions. So there are three major categories of gated channels, which are distinguished as: first, the voltage gated channels. So for the voltage gated channels, these are conformational state that depends on the difference in the ionic charge on the two sides of a membrane. So pag voltage gated channels, again, there's always an incorporation of the voltage of any ions. So electrolytes. Okay. And then next we have the ligand gated channels. So this is a conformational state that depends on the binding of a specific molecule or the ligand, which is usually not the solute that passes through the channel. So etong mga ligand na to, they tend to stick to the integral protein, and then once integral protein or the channel um, reacts to it, it will open the channel or it will open the pores for a certain molecule to go in and out. Okay, so sometimes partners yan. Okay, and then next you have the mechanogated channel. So this is a conformational state that depends on the mechanical forces, for example, stretch or tension that are applied to the membrane. So mechano, so meron siyang um, external factor, okay, for it to be able to have um, or to succeed with a diffusion or the opening of these channels. So these are the ion channels of the different gating types. So again, you have the ligand gated. So with this one, there is always this ligand that should stick first for it to be able to assist a certain molecule to get in. Okay, so if this ligand is not, it is not embedded or is not linked to any um, protein within the cell membrane, of course, it will not open and it will not let a certain molecule to get in. Okay? And then for voltage gated channel, again, there should always be this ion or this protein that should stick within the intracellular space of the cell membrane to be able to have, a certain molecule to go in and out. Okay. And then next we have the mechanically gated. So it can be pressure, it can be tension that would allow the opening of these gated channels. And then next we also have the always open channels. So again, there are channels or proteins within the cell or within the cell membrane that is always open uh, all the time. But then again, these open channels would sometimes do have the specificity of which type of substance they can assist. Okay. So structure of functions of potassium ion channels. So in here, Roderick McKinnell et al. From the Rockefeller University provided the first atomic resolution image of an ion channel protein from a bacterial potassium ion channel called KCSA. So this is the picture, or this is the representation that um, they have produced. Okay. And then example of eukaryotic voltage gated channels are of course the potassium ion channels, and the genes encoding a variety of distinct voltage gated potassium or Kv channels have been isolated in the inner molecular anatomy being scrutinized. Okay. Next, for the facilitated diffusion. So diffusion, during which substances bind selectively to a membrane spanning protein to facilitate the diffusion, and which facilitates or which is being supported by the facilitative transporter. So for example, the glucose transporter. Okay, so glucose is body's primary source of direct energy, and most mammalian cells have a membrane protein that facilitates the diffusion of glucose from the bloodstream into the cell. So an increase in blood glucose levels triggers insulin secretion by the pancreas, which stimulates glucose uptake into various target cells, most notably your skeletal muscle cells, adipocytes, or fat cells. Okay? And then for the active transport, so this is just a review of what I've already discussed. So many ion gradients are very strong and are generated by the expenditure of energy. Hence, we use active transport. So for example, we have potassium inside the mammalian cells that are at least 100 millimoles. So outside is around 5 millimoles. So there's the urge of these ions to leave the cell. Okay? And then for sodium, around 150 millimoles outside the cell, inside is around 10 to 20. So sodium ions wants to enter the cell. And then for calcium, cytosol concentration 10 to the negative 7 moles outside in the cytosol, and then outside around 10,000 times higher than the inside of the cell. So there is an even greater difference than with sodium or potassium. So establishment of such steep gradients depends on the integral proteins that selectively binds the solute and move it against a gradient going through endergonic process. And proteins that carry out active transports are often called the pumps. So next, coupling active transport to ATP hydrolysis. So the sodium potassium pump only in animals, they are only found in animals, and primary means to maintain cell volume and establish the steep gradients needed for nerve muscle impulses. And then other ion transport system, there is no high resolution structure for the sodium potassium ATPase, but the 3D structure of the related P-type pump calcium ATPase is instructive. So there are also different or numerous ion transport systems found in your cell membranes. And then utilizing light energy for active ion transport. This has already been used with the Halobacterium salinarium, or previously Halobacterium halobium, an archaebacterium that lives in extremely salty environments. And then co-transport, this is the coupling activity transport to existing ion gradient. So when you say co-transport, ito yung sinasabi ko sa inyo kanina, that sometimes there should be two factors that would affect the transport system of a certain molecule. So for co-transporting, you have, it can be the combination of your proteins plus ATP. It can also be proteins um, plus your other proteins or chemical receptors. So those are the co-transport systems. So types of transport system, you have uniports versus co-transport system, or symports and antiports. So for the uniport system, this is the transport of one solute at a time. For co-transport system, there can be one solute move across membrane at the same time by a single transport molecule. So two transported solutes move in the same direction is what you call a symport. And if the transported solutes move in opposite directions, it's what you call the antiport. Okay? So, membrane potentials and nerve impulses. All organisms possess the ability to respond to external stimulation, a property referred to as irritability. And it is a function of the cell membrane and seen in nerve cells and amoebae, which can respond to stimulus or stimuli. So nerve cells or the neurons are specialized for the collection, conduction, and transmission of information, which is coded in the form of fast-moving electrical impulses. And then voltage or electrical potential difference can exist between inside and outside of the cell. A positive ion exists on one side of the membrane and the negative ion exists on the other. And then action potential, our present knowledge of the membrane potentials and nerve impulses rest on the work on a giant squid axon in the late 1940s and early 1950s by Alan Hodgkin, Andrew Huxley, and Bernard Katz. So again, with the action potential, guys, di ba na-explain ko na to sa inyo that action potential is when a certain activity is, going on a height coming from a resting position state. Okay. So these are the parts of the neuron with the functions. So just um, study the parts and the functions of this one. But I would like to reiterate that with the process of nerve impulses, mas mabilis talagang or the movement of the impulses are faster when the axon is myelinated. Diba? With the myelin sheath, this part, when there's a myelin sheath, the process of the nerve impulses are faster than non-myelinated. Okay? So speed impulse can travel depends on the following. First, the diameter of the axon. If larger, there is less resistance to local current flow and action potential is easier to propagate. And then during vertebrate evolution, an increase in conduction velocity was achieved when the axon became wrapped in myelin sheath. And importance of myelination is dramatically illustrated by multiple sclerosis, a disease associated with deterioration of the myelin sheath that surrounds axons in the various parts of the nervous system. So once you have this multiple sclerosis, diba guys, you don't have the capability of really feeling. Kaya nga nagkaroon, diba, before ng challenge yung ice bucket challenge. Kasi more likely the impulses are not, traveling in the body normally. Okay? So neurotransmission is the jumping of the synaptic cleft. So neurons are linked with their target cells as specialized junctions or synapses. So no direct contact. They are separated from each other by narrow gap of around 20 to 50 nanometers. So gap is called the synaptic cleft. So with myelinated axons, usually what we know about its the movement of the impulse is that they are jumping, 'di ba? Tumatalon kaya mas mabilis yung impulse. And then synaptic plasticity or synapses are more than just connecting sites between adjacent neurons. They are key determinants in the routing of impulses through nervous system, and their study is very important. Okay, so this actually ends my lecture for the cell membrane biology. Do you have any questions?